All right, and it's 10 o'clock, so we're gonna get started because this is our last class before test. Some reminders, chapter 8 homework is due today. The box is up here with a stapler. My daughter is sick for the third time this school year. Woke up with a fever of 101.4, so I'm going home half the class, but I'm happy to meet with you virtually during office hours if you want to. So if you want to do that, just schedule a time with me and I will send you a Google Meet invitation and we can meet over Google, bringing back fond memories of the that you have in virtual school too. Your test is chapter 68 is on Wednesday. Here, in terms of format, I would expect it to look pretty similar to the last one. I don't know if there's any matching yet or not because I'm not done writing it yet, but there will be multiple choice, there will be diagrams, short answer kind of things. So format wise, it should be pretty similar. Your lab exam is on Thursday. Make sure you sign up for a time. I think there's a review sheet posted for you. Lab exam is very different than any class exam, so I hope Ash should talk to you a little bit about that. Formatting wise, yes, maybe. What do you mean? There should be, I think I didn't look at their page. I'm assuming there's stuff posted. Is it even signed up for a time slot? Yes, okay. There's stuff posted. Okay. And then something you will not forget, which is that there's a conference next Monday, so there's no class. No one ever misses an announcement that there is no class. They might miss an announcement that, hey, something is due or something like that, but no one ever misses that announcement. So next Monday, you are supposed to be going to sessions, which I'm sure you're hearing about, and you're supposed to sign up for those sessions on the Chapel app, I think. So that is what you're supposed to be doing next Monday. Okay, questions about any of that? Nope. Okay. Then we are going to wrap up chapter 8 today. We got partway through, about halfway through, I think, on Friday. We were talking about on Friday, we were talking about different kinds of reactions, the exergonic and endergonic reactions, and the amount of energy of the reactants versus the products, and the amount of free energy change and the energy input or energy output. So we're going to continue along those lines. I have nothing fun for today. I was disappointed in myself. There's nothing fun today. It's just reactions and energy and that kind of thing. So when we talk about reactions and endergonic versus exergonic reactions, we have to talk about the amount of energy that is required to make a reaction happen, which is the activation energy. So this is just the amount of energy that it takes to make a reaction happen. I think about this a lot just in life. You know, I'm at home and I have a task that I have to complete, and it sometimes takes a lot of activation energy to get me to start the task. But once you get started, it kind of goes downhill. Grading, I feel the same way. I sit at my desk and look at the pile of papers and think about the amount of activation energy it's going to take me to be interested in starting this task. When we're talking about reaction happening, again, thinking about this is energy that's needed to alter chemical bonds, whether we're making chemical bonds or breaking chemical bonds. How much energy does it take to make or break those bonds? So this is showing us an example of a reaction. We have A bonded to B, and C bonded to D. So these are what in the reaction? What goes into the reaction? Reactants. So these are reactants. And over here, we're making A bonded to C and B bonded to D. So we're switching switching the way they are bonded to each other. We had A bonded to B, now A bonded is bonded to C. These are what? These are the products of the reaction. So in terms of energy, which one has more energy? The reactants or the products? The reactants have more energy, so what kind of reaction is this? This is an exergonic reaction. So again, we've moved around, we've changed these bonds, we've broken some bonds and made some new bonds. These up here, you'll see have these wiggly lines in between them. These represent what we call the transition state in a reaction. So these reactants are absorbing energy from their surrounding because it's going to take some energy to make it happen. And we get to this transition state, these molecules, because they've got these wiggly lines to represent the fact that they are more unstable. They're not as stable, so they're kind of bent or contorted. They're unstable forms of those molecules. And then that's what makes this a downhill reaction. So we're getting the energy to get to the top of here, and then it's downhill from there. This arrow is representing the activation energy. So this is the amount of energy that is needed to make this reaction happen. Because it's an exergonic reaction, is it spontaneous or not spontaneous? Somebody said it. It's a spontaneous reaction. So in this case, our delta G, I don't know why that happened. Oops. Someday I will master technology. Our delta G in this case is negative. It's less than 0. So this makes it a spontaneous reaction. So take a minute. Oh, there. Exergonic reactions are spontaneous. So take a minute and in your notes, I want you to draw what the opposite would look like. If it was an endergonic reaction, what would that graph look like? So same axes. Okay, now you're all too quiet. Very quietly draw what this reaction will be. So if it's an endergonic reaction, how do the products and reactants compare to each other in terms of the energy that they have? How do products compare to the reactants? The products have more energy, so we're going to put them up higher. So our graph, instead of going up and then down, is going to start lower, and then it's going to go up. So our reactants are here. Our products are up here. And this line is going to represent the activation energy. In this case, because it's endergonic, this means it has to be an input of energy from somewhere else. Somewhere else I'm indicating by a question mark. It has to come from somewhere. It can come from different places. In this case, our delta G, the change in free energy, is greater than or equal to 0, so it's not a spontaneous reaction. So it takes energy coming from somewhere else to make it happen. Remember I said on Friday, what is the answer to every question in biology? Enzymes? Okay, so what does an enzyme do to this? Why are enzymes so important? Why are they the answer to every question?
Speed up the reaction by making the activation energy lower.
Yeah, so they have the ability to actually lower the amount of energy that is needed for activation energy. Okay, when I'm approaching a task that I want to do, sometimes chocolate is like an enzyme for me. It lowers the activation energy. If I eat this chocolate, then I will motivate myself, or sometimes I make myself wait till after I accomplish my task. But so this is showing you the reaction without an enzyme. And you can see this black arrow is representing the activation energy for that reaction. So it's still happening. It's not that it doesn't happen. But if we add an enzyme, you can see over here that the amount of energy that's needed to make that reaction happen is smaller. So what's that going to do to the rate of reaction? You don't need as much energy. It's going to happen faster. It's going to happen faster. So this enables a reaction to move more quickly, to happen more quickly. So what is an enzyme? An enzyme is a macromolecule that acts as a catalyst. Most of the time they're proteins, but we can't say they're always proteins. That's why we say macromolecules. So if they're proteins, what organelle is going to make them? What organelle makes proteins? They're made of amino acids. They're polymers of amino acids, but what organelle is going to make them? Ribosomes, yeah. So these can be made in the ribosomes. And just like other proteins, they might be processed in the endoplasmic reticulum, further processed in the Golgi and sent to where they need to go. You'll notice in this is representing our delta G. What is the difference in delta G for a enzyme catalyzed reaction versus a not enzyme catalyzed reaction? What's the difference? Nothing. It doesn't change the amount of free energy. It doesn't change the delta G. So this is the same whether there's an enzyme or not. So it doesn't change the free energy. And the enzyme itself is not used up in the reaction. So enzymes aren't consumed, so they can be reused over and over and over and over to continue making a reaction happen. So having an enzyme, as I said, will help to speed up reactions. Or it can help to control which reactions happen. If there's no enzyme, the reaction's not necessarily going to happen at the same rate or at all. Some reactions require enzymes to happen. So this lower activation energy, how does that actually happen? How does an enzyme actually lower the activation energy? So we're gonna come back to that in a minute because we're gonna look at the structure of an enzyme first. So this week in lab, you have a lab exam. Next week in lab, you're gonna be doing a lab with enzymes. So you're gonna extract enzymes from a mushroom, and then you're going to subject those enzymes to different conditions and see how it affects the enzymes. It's kind of a fun lab. It's not my favorite lab, but it's kind of a fun lab. So this is showing you what the structure of an enzyme, what the structure of an enzyme could look like. Again, you can see it's a protein. It's a weird shape. They all look different because enzymes are extremely specific. Enzymes have substrate specificity. So usually an enzyme only works for one reaction. Or one substrate. So what is the substrate? The substrates are just the molecules that are involved in the reaction. So they're the reactants. In a chemical reaction. So if it only works for one reaction, how many enzymes do you think are present in the human body? If you need a lot, what's the correct answer? Yes. The real correct answer we don't necessarily know, but what do you think our best estimate is? If you need an enzyme for every reaction that happens in your body, it is not that much. Not as high as 20 million. Nope, it's not even over a million. You guys have really broad ideas of how. Less than 200,000. Less than 100,000. This is making it sound like it's not that impressive because we started so high. No, more than 200. More than 1,000. Okay, so we're between 1,000 and 100,000. More than 10,000. More than 20,000. More than 50.
80,000.
For more than 50, less than 100. Okay, we don't have an exact number at this time. It's about 75,000 enzymes that we know of in your body. Okay, that's 75,000 different proteins, or some of them are not proteins. But all of those have to be made in order for your body to function. 75,000 different ones that your body knows how to make. And you have no idea what they even are. Sometimes I get a name for things like this. 75,000 enzymes made out of, for the most part, 20 amino acids combined in different ways to make 75,000 different enzymes that you need. I think that's amazing. I hope you're amazed. I can see the amazement on your face. You're all amazed. Again, that's an approximate number. We don't know the exact one. So what happens when we have an enzyme coming in, an enzyme present and a substrate coming in, is we get this binding here. So that enzyme, this substrate will fit into that enzyme, and that's the only substrate that will fit into the enzyme. That says enzyme. And that forms an enzyme substrate complex. So the shape of the enzyme is altered a little bit. You can see it's kind of like closing in on it. So it has some flexibility in shape. It closes in with that substrate. And this is kind of what it looks like on a less molecular level over here. So we have two different products coming in. We'll say that those are A and B. So these substrates come in to this area, which is what we call the active site. I don't think I said that yet. The active site is where the reaction is going to happen. That's where the substrate's going to fit in. So these substrates will enter the active site. This is a reaction where two substrates are coming in together to the active site. And now we have the enzyme substrate complex. I don't know why I can't see that. right? After how many years? So stumble on it. Enzyme substrate complex. So they're held in place. They're held into the active site with weak interactions. So that could be things like van der Waals forces, hydrogen bonds, that kind of thing is going to hold them in place. After everything's in there, that's when the reaction actually happens. So the substrates are converted to products. And you can see we have a stunning example of red and green turn to yellow and blue. In your body, they're not color coded in that way. But that conversion, again, is involving bonds being formed, bonds breaking and reforming, and we get the products being released. That's 4. And now they're yellow and blue. They're shaped a little bit differently because they underwent chemical transformations in there. And now the active site is ready to be it's open and ready to be used again, because again, the enzyme is not used up. So new substrates can come in and this can happen over and over and over and over again. Somewhere I have a number. Where's my number? I wrote down a number. I can't wow you with the number if I can't find it. Oh, never mind. It's related to ATP. It can happen really fast. And again, this enzyme has this flexibility to change its shape to hold things in place until the reaction happens, and then it releases it and can be reused again and again and again. So I think sometimes it's helpful to see an example. So a really simple reaction that is catalyzed by an enzyme is the reaction of water and sucrose. What is sucrose? OS tells us it's what? Sugar? What sugar is it? Table sugar. So this is just regular old sugar. It's a disaccharide, C11H22O11. And it's broken down by an enzyme. So that OS tells us it's a sugar, and the ACE is going to tell us it's an enzyme. Oops. The enzyme is called sucrase. A lot of times the enzyme name tells you something about it, not always. But again, that ACE ending tells us it's an enzyme, and the sucro part tells us it's breaking down sucrose. That reaction of water and sucrose that's catalyzed by sucrase is going to produce fructose and glucose. Both of those are C6H12O6 molecules. It's a very simple reaction. Sucrase is the enzyme. Here's water and sucrose coming in, and we produce fructose and glucose coming out. And this could be sucrase. 75,000 different ways your body can do that. Okay, so I said we're going to come back to this idea of how do the enzymes actually lower the activation energy? How does having a place for the reaction to happen actually make it happen faster? So there's actually 4 ways that an enzyme can make a reaction happen faster or lower that activation energy. One is just orienting molecules so that they're in the right position to make the reaction happen. So in order to break those bonds and make those bonds in the right way, they have to be lined up in the right way. So that enzyme having that structure that is unique to only those substrates fitting can cause the proper orientation to make the reaction happen faster. So that's one way to lower the activation energy, is to give it space to orient correctly to make it happen. A second way that enzymes can lower the activation energy is by actually inducing those transition states. So those transition states are the molecules that are a little bit bent or contorted, so they're not as stable. So if they fit into the enzyme in a way that makes them bend or contort, that can make the reaction happen faster. So actually changing the physical structure of the molecule when it binds with the enzyme can induce that transition state to make things just proceed in the way that they should. Faster. The third way that they can actually make a reaction happen faster and better is by making a microenvironment. That has the unique characteristics that are needed to make that reaction proceed. So remember when we were talking about cells, we said one thing about cells is they have these membranes, these membrane-bound organelles that can create a unique environment for something to happen. Like lysosomes often have a low pH because the digestive enzymes function better at a low pH. An active site in an enzyme can be like that. It can provide a really unique microenvironment like a low pH. If there's a lot of acidic side chains that are in that amino acid, and the amino acids that make up that enzyme, the active site itself can actually have like a lower pH or a higher pH. So the side chains that are in the enzyme's active site can influence the environment enough to create a unique environment that allows a reaction to happen more easily and lowers that activation energy. And the fourth way is just by the way that the side chains interact. So the side chains in that amino acid, from the amino acids in the enzyme can actually interact in ways that make the reaction happen faster itself. So all four of these are ways that an enzyme can lower the activation energy. So again, their structure is determining their function. We will talk about that all semester. We'll talk about it all next semester. Structure determines function. So the structure of the enzyme determines its function. And the structure is determined a lot by its chemical formula and side chains. So enzymes in an ideal environment, enzymes lower the activation energy and make everything happen faster or control what is happening. Sometimes some different factors can affect enzyme activity. And this is one of the things you'll look at in lab next week. Factors that affect enzyme activity. There's 3 ones that we always talk about. What do you think? What can affect an enzyme's activity? Temperature. So there are always, you can also think of this as enzymes also have an optimal condition. Remember, we talked about all living things maintain homeostasis, things within a certain range. Enzymes have a certain range of temperature where they work the best. In general, as temperature increases, what happens to reaction rate? It's also going to go up to a point. Okay, that's true with or without an enzyme. If you throw a reaction, try to run a reaction at 25 degrees Celsius versus 37 degrees Celsius, it's going to go faster at 37. Why? What happens as you increase the temperature? Right, everything is moving around faster, so you're more likely to have an effective collision that makes the reaction happen. So just in general, temperature increases will increase reaction rates. But this is also true with enzymes. The problem is if you get too hot, you start to denature the enzyme. So remember protein denaturation? You can break down the structure of the protein. So if it gets too hot, enzymes can't function and reactions will happen well. So enzymes have an ideal range. What do you think the ideal range of enzyme function in your body is? Yeah, 98.6-ish. Okay, or 37 degrees Celsius. That's what the optimal temperature is for your body enzymes. My daughter's temperature was 101.4. Okay, when it gets too high, you start to worry about the fact that proteins are denaturing and things aren't going to function right. I'm not worried about 101.4. But that's one of the reasons that you are concerned about a high temperature, is because it can cause the difficulty for your body to function, enzymes to function, reactions to work. As temperature decreases, what happens to reaction rate? It's also going to go down. But again, there's some ideal temperature. So for human enzymes, the ideal temperature is 37 degrees Celsius. Next week, you're going to extract enzymes from a mushroom. You're extracting sylobiase from a mushroom. You think a mushroom's ideal enzyme function temperature is 37 degrees Celsius? Unless they live in the tropics? No. Okay, a mushroom's not going to function ideally. So the enzyme that you're going to work with next week does not function ideally. We work with bacteria in micro lab. I'm growing a bacteria right now. Its happiest temperature is 55 degrees Celsius, which is really, really, really hot. I don't know the Fahrenheit conversion. It's really hot. That's where it works the best. Guess how much competition there is if you want to grow at 55 degrees Celsius? Not much, because nobody else can do it. So bacteria, well, it's one way that bacteria can specialize and reduce competition is by having enzymes that function at really high temperatures or really low temperatures. There's bacteria that would grow on ice. Okay, again, not a lot of competition for anything there. So temperature has an ideal range and can affect enzyme function. What else? pH. And we've kind of alluded to this, talking about like your enzymes in your stomach function at best at a really low pH, like 2. Your enzymes in your intestines function at a really high pH, 8.5. So having an ideal range doesn't necessarily mean a normal range, but usually the ideal range is 6 to 8 for most enzymes. And again, if you get too high, so too basic or too low because you're too acidic, that will denature the structure of the enzyme, making it not function. No structure, no function. So it's usually 6 to 8, but again, it depends on the environment. There are things that live in weird places, and there are enzymes in your body that function at really extreme pHs because of digestion. So ideal ranges for pH. The third one is concentration. And when we talk about concentration, we could mean the concentration of substrate. Obviously, if you don't have very much substrate, it doesn't matter how much enzyme you have, because there's nothing to make the reaction happen anyway. So substrate concentration or enzyme concentration can affect reaction rates. It's not really affecting the enzyme, but it's affecting reaction rate. So next week in lab, you're going to test pH. So you're going to test the enzyme that you're working with under different pHs to see how it affects the rate of reaction and how effective that is. In addition to these like ideal ranges, there are other things that can affect enzyme activity or make enzymes function better or not better. There are molecules that are called cofactors that are like little friendly little molecules that just help out. They're not proteins, but they're there to help make reactions happen by helping the enzyme itself. These can be organic or not organic. Anyone know any examples of a cofactor? This morning I got up and I live in Western New York where it's gray and dreary all the time. So what do I take? Vitamin D. Okay, a lot of vitamins are cofactors. That's why you need them, because they help to make the enzymes function. And the enzyme can't function without that cofactor. It can't function effectively without that cofactor. That's why you need to eat fruits and vegetables. Because artificial vitamins aren't as good. So eat some fruits and vegetables. There also can be inorganic ones. Things like zinc, copper, those kinds of ions can function also as cofactors for enzymes. If they are organic, they're called coenzymes and not cofactors. So those obviously work to help increase enzyme function. There are also molecules that work to decrease enzyme function. Their name is a little more clear in that they are inhibitors. So they are going to stop the enzyme from working as well. And inhibitors can be temporary, where they bond temporarily, and they're reversible, or sometimes they are irreversible, meaning they bond permanently and permanently deactivate that enzyme. So these will bond to an enzyme with a covalent bond. Wow, I don't know what that word is. I just made up a word. I think I combined elements, an enzyme and covalent. Enzymins, I don't know. So they bond with a covalent bond, and it can happen in two ways. We can have what we call competitive inhibition. Which sounds like what it is. So you've got a substrate trying to come in, and then this inhibitor is competitive. It's shaped the same as the substrate itself. So it competes, and it will bind instead of the substrate. So it looks like the substrate, and it can bind with that active site. So it blocks the substrate from binding. Does that make sense? It's competing with the substrate, so it looks like the substrate. It's mimicking the substrate and can bond just like so that's considered competitive because it's blocking the actual substrate. The other kind is non-competitive. What do you think is going on in this picture? So this is our substrate in both pictures. And this is our competitive inhibitor. So here we've got the substrate, we've got the active site. Is the inhibitor blocking it? No, says two people. Okay, the inhibitor is not blocking it. So this is the inhibitor down here. So why is that called non-competitive? It's not preventing the substrate from binding, but what it's doing? It's binding somewhere else to the enzyme and changing the enzyme shape. And in changing the enzyme shape, it's changing the shape of the active site. So that means that now that substrate, which is specific to that active site, can't bind to the active site. So it's not competing directly with it. It's like coming in from the back and changing the active site so that it can't bind. So if you think of enzymes as locks and keys, this is the robber that comes into the house and changes the lock so you can't get in your own house. I don't know. You guys are all tired and looking at me like, I don't know what you're looking at me. Expressionless. Okay, so they're both preventing the enzyme from being functional. It's just competitive versus non-competitive. And you can also have, and there's no picture of this, sorry. Molecules that are activators. Oh, I wanted to say one more thing up here. Some examples of things that are inhibitors, especially in living things like humans or insects. Pesticides can act as inhibitors. Toxins, things like cyanide can act as inhibitors. Poisons are inhibitors. And a lot of antibiotics are actually inhibitors too. So there are antibiotics that will prevent the cell from being able to make its cell wall, a bacterial cell from being able to make its cell wall. If it can't make the cell wall, it can't survive. So not all inhibitors are bad. We appreciate those functions of those inhibitors. So activators obviously can also affect the enzyme, and they're used to regulate enzyme activity. There's a couple examples in the book, but we're going to talk really quickly about allosteric activation. Which is what you see here. So this is an enzyme. And it's an allosteric enzyme. It's got 4 subunits. So each one of these little openings is an active site. So it has 4 active sites. But then it also has these spaces in here. And these are regulatory sites. So it has these 4 regulatory sites also. So this is an active form of the enzyme, but we can introduce these activator molecules into the regulation sites, the regulatory sites, regulatory. And it makes a more stable enzyme. So you can introduce even 4 of these into those active enzyme sites, the regulatory sites, and that can make the enzyme more stable. When it's more stable, it functions better. So it can activate it. So it's binding in one site, kind of like a non-competitive inhibitor. It binds in one site, but it affects the function at a different site. So you can have conflicting molecules, activators and inhibitors, both working to regulate the rate of reaction or regulate the enzyme. Okay, this is our last slide. And I'll be honest with you, I don't know why this is the last slide. Because it comes from concept 8.3, and we've already talked about 8.4, but this is the way the PowerPoint was given to me, and I didn't pay enough attention to notice it was out of order ahead of time. So the idea of cellular work, we've kind of already alluded to it. There's 3 kinds of cellular work, but we've talked about ATP, but we haven't really talked about ATP. Cells can do 3 kinds of work. They can do chemical work. So this is endergonic reactions, making bonds. they can also do transport work. Meaning transporting things across the membrane against the gradient. Remember, it's not work because it's passive if it's going with the gradient. So against that concentration gradient, so meaning we're going from low to high concentrations. That doesn't happen normally. So that takes work. And the third kind of work they can do is mechanical work, which is like actually moving stuff, like cilia or flagella or muscle cells contracting or moving chromosomes during cell division. So this is actual causing movement. All of these need some kind of fuel, and that fuel comes from the molecule ATP, which we've talked about, but not really talked about. So ATP, yeah. Endergonic. You're welcome. Does this structure look familiar at all to you? We've got a house. We've got a driveway. We've got 3 pools. This is a really nice house. It's got 3 pools. Okay, this is adenosine triphosphate. So adenosine is a nitrogenous base. This is ribose, which is a sugar found in, where do we find ribose? Somebody said something. It's in one of the nucleic acids, RNA. So this is the sugar found in RNA, and this is triphosphate because there's 3 phosphate groups attached to each other. This is cellular fuel here, and how that happens is through the hydrolysis reaction. So ATP hydrolysis is what releases the energy. Where is the energy stored in this molecule? Where do we store energy in molecules? In the chemical bonds. So the bonds, there's lots and lots of bonds in this, obviously, but the bonds over here in the phosphate, the triphosphate area, are more rich in energy because they all have these charges on them. You see a lot of negative charges. That's like a spring that's compressed. Okay, that negative charge is giving a lot more energy. And so we can hydrolyze this, meaning we can add water, which you can't see because that's a bad color choice. We can add water, and we're going to break one of those P to P bonds. So we're going to release a phosphate group, and this is going to be now ADP, adenosine diphosphate, because now we only have 2 pools in our house. But that releases energy. It releases specifically 7.3 kilocalories of energy per ATP molecule, which doesn't necessarily sound like a whole bunch. That's under standard temperature and pressure. In your body, it's up to 13 kilocalories per ATP, which again, doesn't sound like a whole bunch. 13 calories, that's like what? Like 2 Tic Tacs or something? Okay, 6 Tic Tacs, I think. Not a lot of energy, but if you think about this process happening over and over, a muscle cell in your body, one muscle cell, can go through this process at a rate of 10 million ATPs per second. So that's a lot of kilocalories. Okay, this is also a reversible process where we can put them back together. We have a dehydration reaction that can be used to take ADP to make ATP again. So this is a cyclical reaction that can renew itself. This is a spontaneous reaction on this side that's releasing energy. On this side, to add that phosphate back on, it's not spontaneous. We have to add energy to make it happen, but it's still kind of worth it for your body to do that because it can produce a lot of energy. And this is the active transport that we've talked about before. The ATP allows this protein to change shape to move these solutes from low concentration to high concentration. So that's transport work. Okay, I'll see you on Wednesday. I had some study things, but you all know these things.